Hi, my name is Julio Jaramillo and I'm going to talk about the Dermatan sulfate epimerase 1 expression and its localization may interference with dermatan sulfate synthesis and breast cancer cell growth. So, before starting, I'm going to do an introduction about the breast cancer and the molecules that intervene in these processes. So, the breast cancer is one of the most important and prevalent types of tumors worldwide. In addition to this, the incidence has increased, and by 2050, many more new cases are expected. These type of tumors can be categorized according to different parameters, such as histological, that takes into account cell proliferation and mitotic counts. According to the morphology, infiltrating lobular and tubular carcinomas are present. And according to the molecular parameters, there is talk about the presence or absence of estrogen and progesterone receptor. Within the cellular processes, different substances are present which has been studied in this article, which can determine the cell growth and proliferation of these tumoral cells. Among these we find the proteoglycans, which present covalent bonds with the glucosaminoglycans. And these are important in all the part that has to do with vascularization and cancer metastasis. The main glucosaminoglycans that take into account is chondroitin sulfate, which is present in the extracellular matrix, and dermatan sulfate, which differ from the previous one due to the presence on an epimer A2A. For the cellular behavior, the synthesis of the last glucosaminoglycan must be taken into account from dermatan epimerase, where the high values of these in tumor cells are related to the higher growth rate. The objective of this article was mainly to investigate the expression of Dsepi1 in the multiple breast cancer cell lines and its amplification in cancer progression. So, I'm going to talk about some different materials and methods, and one of them is MTT assay, which is a test that indicates the viability of the cells and their metabolism. It is mainly based on the fact that a purple substance is applied to the cell and which is metabolized by the enzyme oxidoreductase that is present in the cytoplasm of the cells. In the study, they incubate the cell for 24, 42, and 72 hours, although sometimes at 96 hours. The other method is RNA extraction and real-time quantitative reverse transcription PCR. This test is responsible for making a retrotranscription of a strain of RNA complementary to a strain of DNA using reverse transcription. It's used to detect for the quantification of gene expression, molecular detection of genes, and the study of the genome of DNA virus. Other method is flow cytometry staining and data collation. The study is responsible for reading data using light that crosses the cell that are in suspension in an aligning manner. It is mainly used to monitoring DNA content, phenotypic expression, proliferation, apoptosis, and among of other requirements. In addition to this, the study was used to compare flow cytometry data between cell lines, and histograms were plotted for visual evaluations. And the last method that we are going to talk in this article is immunofluorescence staining and confocal imaging. This technique is developed from the binding of antibodies that are bound to a fluorescent substance that determines the presence of a molecule. This technique is mainly used to analyze the presence and distribution of proteins, and this technique, a confocal microscopy, was also used for imaging acquisition. So, in this part, I'm going to talk the results, and this picture talk about the ASCAP-BR3A and ASCAP-BR3M light cells. In this point, we can see that the cell extract and the medium, the protein dermatan sulfate, are expressed, but in the ASCAP-BR3A is only expressed in the cellular extract but not in the medium. And now, this result shows how MCF7 and ASCAP-BR3M have a larger cell population. This blue growth refers to the control, and this red group refers to the lines present the experimental cells. So we can see how in MCF7 the Dsepi1 is present, but in the SKBR3M is not expressed. This result shows the staining in the cell to determine the places where the enzyme is located in the different cell lines. For example, in this we can see the Dsepi1 is located in the nuclear area. In this is located to the cytoplasm in different parts. And this we can see is located in the cytoskeleton. And then it is a combination of the three. And now, in this picture, tell us about the distribution that Dsepi1 has in the different cell lines, determining that the SKBR3M has a great distribution near to the Golgi device, and the comparison of the other cell lines found in other regions. For example, in this that is studied, and this shows the Dsepi1 is located in the Golgi apparatus area, in the others are located in different parts of this. And the last one, this picture shows out how the SKBR3M, the expression of Dsepi1 is more to compare the other cell lines like MJ or MCF7. So, the conclusion of the sum of authors, one of them is Dsepi1 has been found to be overexpressed in glioblastomas. That is correct because the different experiments like immunohistochemistry shows the different location and the different sites that express the Dsepi1. These authors determine that in this line cell, the cell's ability to synthesize full chains of CS or DS will affect the cell's viability, motility, and adhesion to matrix substrate. That is correct because in the experiments we can see that the interactive or interview to these glucosaminoglycans in the tumoral effects. And the last one told us the high Dsepi1 expression was also shown to be correlated with increased DS production, and that is correct because the experiment showed us that the cellular that contain more DS or CS, Dermatan sulfate, have more expression of Dsepi1. So the conclusion is this article is so relevant and important because show us the different molecules or glucosaminoglycans interview in the cellular proliferation of the tumoral cell of breast cancer, and we can find a management for this. And then the formation of proteoglycans from Dsepi1 has contributed to the growth of carcinoma cells, and there will be a target for the treatment of these cells. And that's all of my article. Thank you for watching. Subscribe please, give like. Bye.